morning, everybody, and welcome to this webinar. My name is Craig Pensini. I'm with Perth NRM, but also today this webinar is being co-hosted by our RegenWA entity, because that's where we're aligning the project work that's happening with this eDNA technology. So I'm just going to quickly go through an introductory presentation. Before I do that, a little bit of housekeeping. Wonderful to see so many people online, and there will be time at the back end of today's webinar to have some Q&A. I would ask everyone if they could put their Q&A or their questions into the Q&A section at the top of the Teams webinar front. So you'll see the little question mark with Q&A. If you want to raise your hand at some stage to point out a particular question, you can do that as well. We'll endeavour to answer as many questions this morning as we can, but if you put them in the Q&A section, we'll circle back and come back to those questions and see if we can provide you with a suitable answer. Today's presentation is also being recorded, and it will be available shortly after the webinar on our RegenWA website, which we'll post the details for. So thank you once again. So just quickly, I will go through an introduction presentation as to the background of today's webinar. So we're just waiting. Here we go. So the today's title of the webinar is eDNA sampling and how to get a good result. So this is a follow-on from a workshop that we ran earlier this year, back in May, actually, the beginning of May, around eDNA. We had a workshop out on a property in Western Australia, and this is in the context of the larger Landcare Australia, Landcare Agriculture Biodiversity in Action project, which is actually funding this work and the ongoing sampling, etc. So just quickly, we'll go to the next slide, and just a quick agenda for today. So intro today, we'll then go to our two guest speakers this morning, Marina and Shane. They'll introduce themselves. We've got a very short collage of videos just showing some of the sampling that was done last year in spring on our properties out here in Western Australia. We've also got, for your viewing pleasure this morning, some of the sampling tools, and we can just quickly show a bit of a physical demonstration of how they work, and then, as I said, quick Q&A. So just going on to the next slide. So a bit of a project background and overview. So this project, as I said, environmental eDNA, is to detect and prioritise conservation efforts for threatened native species across 3 properties or farms in Western Australia in the regions of the Magumba and Bindoon area. And our goals are to try and improve farmers' knowledge and confidence, identify and protect any potential threatened or endangered species, implement targeted management of any actions based on the testing that we do, engage with producers, those farmers and their neighbours, etc., in planning and maybe implementing some of the outcomes that we are able to generate from this technology, and to really build some industry capacity. And hence a bit of why we're doing this webinar today is to make people more aware within the broader industry about how you might want to adopt what the evidence-based actions you can get out of eDNA sampling. So next slide. Very, very quickly, I'm just going to say that, yeah, in the testing that we've done to date, we used a combination of sampling technologies. There were 3 of those. There's different technologies, but the ones we used were the rollers, or the terrestrial sampling, which you'll see today, what's called a water sampling or syringe method, and then an air filter. And as I said, done over 3 properties. Next slide. That previous slide had a picture of the water sampling being done, and this is just a couple of pictures of one hanging a air sampling tool in a tree, and then the roller on the ground taking a terrestrial sample. Okay, so I'd now like to hand over to our presenters today. So to kick off with, Marina de Oliverira is going to start with the presentation. We'll just let the slides come up for that, and I'll allow Marina to introduce herself and explain what she's been doing with eDNA.
Thank you. So good morning, everyone. I am Marina. I am here today with Shane. We are part of two sister groups, distinct but sister groups within Curtin University. Next slide, please. Thank you. So I am Trace and Environmental DNA Lab. Shane is eDNA Frontiers. We work in the research end of eDNA, while Shane works in the applied services. So we do researches focused on water, terrestrial, many things, while eDNA Frontiers does not necessarily researches, but they can process samples as they are presented. They don't need to have a specific research questions. You can just have a set of samples that you want to analyze and give to them, and they will do it. While we need to have a big research project behind what we do. Correct? So the next slide is what is eDNA. So I don't want to scare you, but eDNA is everywhere. If you look around you, you find on the walls, on the table, DNA from humans, your dog, your cat, the chicken and the rice that you had yesterday, the grass that grows outside your house. And that is very good for what we are proposing, which is sample the biodiversity through this eDNA, through this DNA loose in the environment that is everywhere. Next slide, please. So eDNA, environmental DNA, is the DNA deposited by living or dead organisms, whatever they live. They can come from things that you can see, or for things that are difficult to find or are very tiny. Can you please go back to the last slide, just so I would like to point to things about DNA. So although DNA is always the same, so the A, T, C's, and G's that make the DNA, they're always the same. Where they come from makes them have different characteristics. So first thing is abundance and size of the organism. So if we look at whales, there are not many whales in the sea, but they're very big, so they release a lot of DNA. But if you look at plankton, they're very tiny, but if you sum the weight of all the plankton in the ocean, you see that they weigh more than the whales. So they are tiny, but they have more abundance. So that's one thing, the size and abundance of the organisms. And the other thing is the shedding rate of DNA and the behavior of animals and plants and organisms in general. So a few things, they shed less DNA than others. So if you think about insects or reptiles, especially lizards, they shed less DNA compared to other vertebrates like mammals and birds. And that's important for what we do, because the way they shed DNA and where they shed DNA and how much DNA they shed will impact directly on how we sample and how we find these organisms. Next slide, please. Yes. So this DNA that is released in the environment, it can be captured from environmental samples. It can be water, it can be sediment from the bottom of a river, it could be soil, it can be air, it can be anything. All the species, they have unique DNA profiles, and these unique DNA profiles, they are in this mix of heterogeneous DNA in environmental samples, and we want to pull those sequences from that sample and identify that. And this technique is relatively new, which means that we're still evolving and learning. A few things are better established, but we're still developing new tools like airborne DNA, which is a new tool, so very exciting field. So there are three main types types of eDNA analysis. Shotgun sequencing is more used in the research stream than the applied services one. What we use most in general is metabar coding and qPCR. So metabar coding is a way that you can see all of the sequences of a group of organisms you're interested. It can be mammals, invertebrates, amphibians, whatever, or a combination of those. While qPCR is a species-specific assay, so we look for specific species like Frontiers Curtin or Divercali or something that you're really interested. They have pros and cons. And the best tool for your question will be context dependent. So if you want to have a broad list of species that happen in your area, you might be better with metabar coding, but it might not be the best tool to detect very rare species because you have a lot of DNA from things that are more abundant. But if you're looking for one specific species and there is an assay for that species, we would go for qPCR. So how does eDNA metabar coding work? So first, there's the release of DNA into the environment by that organism. It can be a plant, bacteria, whatever you're interested. We collect that environmental sample, in this case is water. We filter it so we can concentrate the DNA in the filter. We extract the environmental DNA from that sample. We identify the unique sequences presented in that sample, and then we match that to a database. We can build our own databases or we can use public available databases. So they are built in a way that we get an organism that we know what it is, and then we do attach a sequence to that identification by a taxonomist. There are a few practical considerations when we are talking about terrestrial eDNA, which is my field of work mainly. So first thing is the purpose. We have to be clear in what DNA can and cannot do. Next slide, please. Yes. So eDNA is very, 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 very good at detecting presence or absence, but I'll talk about absence in a little bit. But it's very bad at detecting abundance of species because there are many factors that can influence the abundance of DNA we detect. So if you want to answer that is yes or no, I found this, I did not find this, eDNA is very good. The non-detection of eDNA it doesn't mean that the species is absent from the area, but it just might mean that it wasn't there at that time or the methods did not pick up, which is a flaw of any research method. The next thing is research and environment, species and environment. So you have to understand what's your target species and what's their behavior in your site. So if you're looking for a community or a target species list, you also have to consider the seasonality and the behavior of the species. So if you try to sample something just after a big rain, all the DNA in the environment might have been washed away. If you're looking to sample species that has a very narrow home range and likes to live in burrows, but you want to sample a different type of substrate, then you might not find it. It's not because it's not there, it's just because you're not looking exactly where you should. The next thing is sampling design. So we have to match the method to the questions and the setting of the sample, the sampling points. So the number of sites we are sampling, we have to understand the ecology of eDNA. So there are the effects of transport and degradation. That's especially true for airborne DNA and for water DNA, rivers, oceans. So they're transported by currents or wind. Every DNA is degraded at some point. So bacteria like to eat organic matter. It includes DNA. So we can consider a positive detection as something that is relatively recent. We have the field of ancient DNA as well, but that's another topic. So other things that we need to think about is, are the methods going to be repeated over time? And if they are, who is analyzing the data? Is it the same person or different people? Is the method that I'm using today a good method because it's has been validated and taught, or is it because I liked the idea, and then is it going to be a good idea to be repeated in the future? And last thing is, not the last thing, the second last thing is that data quality, that we have to build confidence through replication and controls. That is, the replication means that we need to take many samples in the same area. So we can say that the species that we are looking for is not there, not because we just looked at one sampling point and that sampling point didn't have with DNA. It's just that we try to diversify the places that we were looking for, and in that big area, and those number of samples, we didn't find it. The other things are controls. So there are two types of issues. One is human contamination. So our DNA is everywhere. It's in our clothes, our hands. So between sampling points, it's important that we change gloves. We try not to touch our face, touch our hair, touch our shoes when we have gloves on, especially between sampling points and between sites. And that we have appropriate field controls. So if you are hiring a service like eDNA Frontiers, they will tell you what a good kind of field control it is for your sample type. And the last thing is the practicalities and the permission. So we have to think about the logistics of plan of permissions, ethics. If we are going to sample, I'm going to give an example of nest boxes. So we had to have some permissions from ethics committee because the animals might be there and we were going to disturb them with our sampling. So I brought today two examples in the research context. One is a little one, the other one is bigger. So this community group came to us and they had installed 8 nest boxes, and they wanted to know who was using the nest boxes. Is it the brush-tailed phascogale or is it the red-tailed phascogale? It was in Wandu National Park. So we took a student, we made this as a student project. We took a student to Wandu National Park. We swabbed the entrance of the holes and the outside of the nest boxes, as well as the inside of the nest boxes. Next slide, please. Yes, that's our beautiful student. And we found out that just brush-tailed phascogale were using the nest boxes, all the nest boxes they were being used, which is very cool. So that was a small project that got the results that this community group needed to take their management actions. One other thing that this is also a research project, but this is a big scale research project, is the Living Waters of Western Australia, which is a project led by Dr. Angus Laurie. And the idea is that we have been monitoring water for almost centuries now. And usually we base things on indicator species. So, excellent, please. If you look at some species, we usually do it by sampling with nets or something. When we find some species, we say that's a bad ecosystem condition. When we look at other species that are present, we say it's a good ecosystem condition. It could be snail, shellfish, it could be frogs, fish, anything. But when we talk about eDNA, we don't need to look necessarily at species. We can look at sequences. And one thing is that a lot of sequences, they are not in the database, especially for the little things. So the idea was that we could use eDNA to have those indicator sequences, but not attached to a specific group or specific species. So the research group, so that's the idea, that we are going to move from species to sequences. And there was this large-scale effort, 172 freshwater lakes across southwestern Western Australia. So they did a panel of 14 metaphor coding essays, meaning that each essay was targeting a different group, so mammals, invertebrates, bacteria, fungi, and everything. And the results are that we can link sequences with no species identification to the quality of that water, which is very interesting. And my project specifically now is to try to translate that into terrestrial ecosystems. So that's one other use of eDNA, is not only to look at species presence or absence, but we're also trying to expand it to other uses like monitoring ecosystem condition in a more broad, holistic way. And I talked a lot, and I think that's all from me.
Fantastic, thank you, Marina. So I'm Shane from eDNA Frontiers. So as Marina indicated, there's two groups at Curtin University, and that relationship is really quite special, where we get these things that bounce between research and something that we are able to do is translate that out to the community, government, industries, so that this cool research technique can be used, and it's something that tends to capture the imagination. So I guess the point of my part here is to talk through how to get a good sample result. So if you just go to the next slide, please. So the point that we're really getting at is how to get a useful DNA result. So it needs to be detectable, it needs to be relevant, and it needs to be workable. So when you look at the environment, you need to consider how an animal interacts. And as Marina's already covered, depending on what you're looking at, might mean you look at different sample types. So if you're, water is a really great place where a lot of things end up. You also get the things that are living there. You've got animals that move through the landscape that get bitten by mosquitoes, and they leave their poops and wheeze behind, which are actually really good substrates to find out what's there, and to some degree what the contents are reflect what that predator has been eating as it moves through the environment. And so when you look at that, then there's lots of ways of taking samples. So the next slide talks to all of the work that's been done between, everyone in the community, basically looking at all of these different substrates and trying different ways to capture these samples. So I won't go into that much more, but the next slide talks to the main one, which is water. So it's a really simple way of filtering water. And if you see that picture on the right-hand side there, you've got water of different types between seawater, wetlands, rivers, etc., different colours, but they've also got different things in them. And the simplest way to do this is we've got kits. Ultimately what you're doing is filtering that water onto a membrane. And we've got kits that operate with a syringe with a little connecting filter on the end. And the idea is that you try and push a litre of water through that or thereabouts, and you capture everything on that filter. It's very simple to do. There's a manual way of doing it, and we'll go through some of the other ways to help speed this up a little bit. So these water signals can be very relevant for lots of different targets. The signals that we get out of water tend to be quite abundant, so there's a good way of looking right through the biodiversity in that water sample. They're also quite dynamic. Water seems to keep the bacteria animated, so bacteria is the main thing that breaks down these DNA signals, so they don't last forever. And that's an important part for biomonitoring. You don't really want things to last forever. But water does have that concept of when you've got a flowing river, then things will be flowing down the river. And if you've got a fast-flowing river, then it's going to travel further than a slow-moving river. There's also depth and sides, etc. So there's sides of a lake, for instance. We'll go through some of those a little bit later on. But the other useful thing out of a water sample is it extracts well. So the laboratory processes can work with it really nicely, and you get a good result. So moving on to this next slide, so this has got a couple of animations actually. So for starters, so one of the problems we had in Western Australia is there's not a lot of water around to sample. So we've had to look at what alternatives are. And this actually came out from a similar thing to Marina talking about with woody hollows. All these woody hollows and trees around the landscapes, what's using them. So using swabs, which is an approach to get to, but you're only getting a small surface area. So to increase that surface area, we started using these little paint rollers, and that gets around the outside of woody hollows nicely. The other upside to rollers are that you can cover a large surface area and get a larger surface result. But what we've also found is it can be used on all sorts of different surfaces, like trees and bushes and street signs and all sorts of things, and get a nice big representative signal. So, yep, sorry, can you just go to the animation on that one, on the previous slide?
Yeah.
There should be an animation to press next. There we go. And so some of the other things to sample a terrestrial environment is spiderwebs. So they're nature's biofilter, and showing Josh Newton here, who led this study. Proof of concept was that spiderwebs work really nicely. And they pick up vertebrates really well, but don't get invertebrates, despite the fact they're there to eat vertebrates. I guess the spider's chasing them, but we don't necessarily pick them up so well, and reptiles aren't particularly great. And it's good, but I mean the problem with spiderwebs are that they're not well dispersed in nature. The web architecture is different, the suspension time, various different things. So while they're good, they're an opportunistic sample to take an environment, and they can be quite good if you spot them. Quite often, once you get your eye in, you can see where they are quite a lot. So they're a good sample, but you can't necessarily design a study around them. So the next step, sorry, there's one more. This animation, there we go, was air. So air is a really useful concept to replace water in a dry terrestrial environment. It covers, what we found is that the air samples take a much larger distance in, so we're talking kilometres. So if you get a hit on an air sample, it could have come from anywhere in the surrounding kilometre or two. So it's good for large areas, but if you want a refined result, then air's at the moment is not looking like it's going to be appropriate. But this is something that's a fairly active research area, and we're still drilling into where that fits. But it certainly seems to have a place. We just have to work out how. Okay, so the next one is, yes, so rollers. So yeah, as I mentioned, when there's no or not much water, it gives a really nice broad range of targets. The signals are less localized than air, and they're quite dynamic. They don't stay around forever. And you can get across lots of spaces, and you can pick your areas that are facing the wind direction, or and various. You can tune that somewhat to get samples that are relevant to your environment. You can also sample things anywhere in the area. I've seen people time themselves for about a couple of minutes and just run around getting as many samples as they can. But you also have to be careful of how much inhibitors you pick up in that process. Rolling over dirt, for instance, and making it black is not necessarily helping, because that then is counter-effective for the lab work. Next slide, please. So we've covered some of this, but the main thing is to try and keep clean. So, think CSI forensics, wear your gloves, change them regularly, keep a clean workspace, clean it with detergent, or bleach is better. Bleach breaks down DNA, does a better job of it. Ethanol doesn't kill DNA, so that might stop the bacteria, but it's not going to stop the DNA. So yeah, just clean things and be mindful of where you're putting your hands when you're moving around your sampling process. Try to use a standard method when you're collecting, so come up with your theory of how you're going to open packets and pull out your kits, and when you're going to put on your gloves. Have someone maybe watch you to see if you touch your nose or pull back your hair or something. Also, just be mindful of that site-to-site contamination. Anything that you take from one site to the next site, just be mindful of what's there, because it might be carrying a signal from that last site that you then might pick up in your new site. Controls are super helpful. Everybody should do them. Not everybody does. But water controls are a simple thing like filtering. If you put water from your home or a rain tank or something that's completely unrelated to your site, and take that out to site, open it up, and then filter that on site. That helps you identify where you've got contamination coming from off-target areas. Same thing with the rollers. While you're out there, if you roll yourself as a good control, then you know what you're bringing from the last site and what might turn up in your new site. Either that or something that's something that would indicate that we're bringing some signals from the last site. And they're good to compare against, because anything we find in those controls, we basically delete from the test result when we do your sampling. We'd encourage you to try good labelling without massive long strings of alphanumeric characters. It's very hard, although we do try and get people to put all that information into a website now, so we don't have to try and read everyone's writing. Metadata is also really helpful, so if you see a phascogal walking around, write it down. If you see some damage or impacts to the landscape, then write it down. Those things are super helpful. How much water you've filtered, how much area have you rolled, all of those things. They don't necessarily affect the sampling, but it can help you interpret your data later on. Planning for preservation. We have tried to make all our kits ambient storage now, so through preservation solutions, so you don't have to freeze everything. Soil is still something that's been resistant to preservation methods, so freezing is still best for that. But water and these rollers can work with preservative quite fine. And then monitoring sample integrity, just try not to leave them out in the sun and let them get too hot. Free store cycles is also not something we encourage. And just making sure that they're not fluctuating too unusually from one site to the next. Next slide, please. So when you're picking a spot, remember what the question is that you're trying to ask. If you're after a community result, or if you're trying to work out if there's foxes in the area, that sort of helps direct what your question would be. Always remember that access and safety needs to be paramount. Try and be careful about how you're accessing a high treetop or deep into a water body or something. Just remember that if you need a follow-up method, then you might have to go and do that again in a different season where things might be more dangerous. The site resolution, this is a question that comes up a lot. If you're going to a small wetland, for instance, how do you sample it? And I guess there's a couple of schools of thought there. You can go for the ecological zones around it, like something that's heavily treed, something that's full of reeds, something that's different regions around the outside, versus the representation. That big project that you looked at earlier, the LEWA that Marina presented, they did all of that, but then ended up settling on a representative of 6 replicates from one site at one side of the river, of the wetland. And that was enough to get the information from that whole wetland. Looking at all of those ecological zones didn't necessarily help his purpose, but if you're looking for rare organisms, then those different ecological zones could help. Replication plan is you've got a stochastic with a molecular number concept. Not everything's going to end up in one sample, and so biological replicates are super helpful. We do all of the technical replicates in the lab, so you don't need to worry about that. But at least 3, the gold standard is 6. Also, what's suitable and what's sensible. Sampling does need a reality check every once in a while. Sure, you could filter 50 litres and get a sample and test that, but is that realistic? And so you have to pull it back to what sample you can actually get. Things like water and air movement are a consideration. Typically water is fine from the outsides and in the shallows. You don't want it too shallow, but maybe 50 centimetres or so, a foot below the surface is a good sample. You don't necessarily pick up a great deal more by going right out into the middle and going deep into that lake. Most of the life in that lake is around the outside. Same thing with height when it comes to air. Things get more dilute the higher you go up. Volume and area sampled, more is better, but it doesn't necessarily, that accumulation concept, there's a point where it doesn't add much. And then, as Marina's already mentioned, rain has a big impact. It washes everything out, dilutes everything out, and you get less signals. So try and keep the season that you're sampling consistent between your sampling times. So in terms of how to use these, so I guess this presentation will get shared. So there's a couple of videos you can look at on our website. It just runs through them, and we'll aim to keep adding some more there as we go. And just to finish on the last slide there, just talks to when you get good samples, you can get good data. And so we've got a data scientist that's really worked hard at how to make your data come alive. And so this is basically means getting all of these reports online and allow you to interrogate everything yourself. So If you collect that metadata, like lats and longs, then we can map it. We'll try and get everything that hits a species level, a link, which is the second picture there, through to Wikipedia, through to NCBI, and through to GBIF, and all of the good databases that have heaps of information on it, as well as the taxonomic list, of course. And then some interactive graphics displays like your phylogram and some richness plots, etc. But also we're moving all of the data towards what's called a FAIRY guideline, this concept of making data findable, accessible, interoperable and reproducible, so that all data sets can play with each other happily. And that is the end. Thank you very much for listening.
Excellent.
Oh yes, and just to finish on, I was just going to do a small plug for a webinar that's coming up, which is 3 other researchers that are resident at Curtin. One talking on natural capital accounting, Angus there, who's talking on his living waters of Western Australia, and then Josh who's talking, who has been specialising in eDNA. He's also the guy that did the spiderwebs. Thank you.
Thank you very much, Shane. All right. As I said earlier, we're just going to run a very quick series of audio visuals. So these were taken in the field when we did our sampling in spring of last year. And that'll sort of give you a bit of a flavour for physically what we did. Now, the reason I wanted to put these up is that they weren't, I don't know, 100% technically correct. This was our first time with this technology. So this is the water sample being taken and the sample being pushed through the syringe. As Shane mentioned, about a litre of water is preferable. What you find that even in clear water like that, it gets very difficult very quickly to push that syringe and to get that water through that little filter that's screwed onto the tip of the syringe. Shane will go a bit more of that when we look at some of the. So this is the air sampling kit. It's actually quite simple, probably the easiest to deploy of all of the sampling tools that we used. Basically, it comes out of that sterile package, and then there's a zip clip. You find somewhere at the appropriate height, in this case was a tree, and you adhere it or zip clip it up in the tree. We left ours out there for about 5 or 6 days before we went back and collected or recovered the sampling tab, and then it goes off to the laboratory for processing. So basically it's sitting there capturing the DNA as it's circulating there. And this is the roller. So this is the terrestrial sampling. There's a little bit more involved here in getting the kit set up. So this is actually prepping everything. So this is a solution that goes into a little vial. Yeah, so that goes to one side. Then we reach in and we get our actual roller out, which is in its sterile packaging, and that's got to be attached to the handle. Yeah, so, and we don't want to be touching any of that, even though we've got gloves on. We're going to try to avoid touching that with anything. Then we moisten it up so it is able to grab as much material as possible. Now, I'm going to give points or gold stars to people to work out what we could have done better. This is the very, very, very first kit we did. We did one roll. But what we should have been doing was multiple rolls in multiple areas, which as we went along and we further read our instructions is what we did. But it at least gives you in a short space of time the ability to see how the kit is deployed.
It's a good one, suitable for video.
Yes, suitable for video, exactly. Yeah. Okay, so that's the short videos. So now, Shane, we've seen the presentation from Marina and yourself about these kits. You know, why would you deploy one? What are the best settings? You know, etc. Maybe we can talk a little bit more about the practicalities, because I've been making a couple of notes here of some of the Q&As as they popped up, and thank you, Thomas. He's put them up on the chat. So Maybe as you're talking through that, I can sort through some of the Q&As and we can start looking at answering those too.
Yeah, sure. So where's my hands? Okay, there. Yeah. So yeah, water is a relatively straightforward thing, but it just involves. There we go. I've stuck it up straight up. So I guess the first thing is gloves. So we put a pair of gloves inside this, and largely those gloves are to stop the human from getting in the sample. So depending on what you're looking at, we're most likely going to be testing it for vertebrates. Most people are interested in vertebrates, so we're a vertebrate and we shed a lot. So the main thing is to try and keep us out of the sample. So the idea is to pull out the gloves and put your gloves on. So I won't do that now. But the idea is that you pull out your syringe. This is the syringe that you can use. And then everything's got these little interlocking things to connect the filter onto the syringe. And same thing with the preservative. So that'll come together. But I guess in terms of how you want to start, these syringes have a little coarse and a fine filter on the end. I don't know if you can see that. But the idea being is that you don't want big chunky organisms getting into the sample because they only end up on the filter and dominate the reads. So the idea is to try and filter some of the extra junk out and why you're doing it. So you're obviously sucking up the water, and then you basically, when you're doing it manually, this is how you do it. You pop it on, you squeeze the water through, take it off, take your water sample, put it back on, push it through, and there's a process to doing that. And for a litre, that's like 10 to 15 times. So it's a lot, and you'll find that after about 4 or 5 pushes, depending on what water you've done, it gets really hard.
Yep.
And it's not because you've got little noodle arms. That's how it is.
Yeah, and that's exactly one of the questions that we had. Is that somebody's done some recent sampling and it was too difficult in the sieve to get through. So I think you might have a couple of options there.
So what we switch to, because otherwise you get sore hands, is your old corking gun. Nice and simple. So if you've sucked up your water, then the idea is you can just pop it in there, and then you're just pushing that through, and that makes life a lot easier. And you can push that through much better, and you'll get closer to a litre that way. At some point it's going to get hard, and it won't be able to push any further. Then that's the time to stop.
Yeah.
Ultimately, that means you've got enough stuff on this filter that's going to be readable. The idea when you're collecting that sample is try not to get sediment sucked up from the bottom. I appreciate up in the Pilbara there's a lot of suspended sediment that just can't be gotten rid of. That's okay, but at some point it's going to fill up nonetheless, and it's really hard to get around it. Some wetlands are chock-a-block full of some suspended biological sediment, and in which case these are all 1.2 micron filters, and that's a good size to. I guess we don't necessarily always want bacteria. Bacteria are always going to be there, so that size will let bacteria through, but we'll still capture all the other things. If you know that this place is going to be super heavy, then what we've done is switch to 5 micron filters, and that allows you to push a bit more water through, and that gets a better sampling volume. But I wouldn't necessarily stress about not getting a litre. It's good to get the same amount everywhere, of course, but if your question is just to discover the biodiversity, then any amount will get you there. We've had like hypersaline lakes, whatever reason, salts are really challenging to push through filters. It seems like the crystals block things up really quickly. One of the big international projects looking at the hypersaline lakes is settled on 250 mils, and they tried to get that and couldn't even get that. But the result that we get out of it is still relevant. It's still good. All the abundant stuff is there. It's just some of the rarer stuff falls away. So that's consideration for water sampling.
Did you want to also bring out your
in
green machine?
True tradie theme. The other way is to separate your plunger, and then you've got your filter and your syringe. Oh. And here's one I prepared earlier. So we've got these drill systems. So it's a regular old drill. It's attached to a peristaltic head. You put your filter, the coarse filter, the syringe body, on one end, and then this end is your filter. So these only go on one way, so it's nice and easy to tell. And then you drop that into the water, and you can filter it, and it squirts it through. So in seawater, that'll push through about a litre in about a minute, so it's nice and quick. In fact, you keep going until it's blocked effectively, and sometimes. But when something's really chock-a-block full of sediment, this will still clog up and stop. And the beauty of these drills, this is an impact driver, so once it gets too hard to push it, it'll just start clapping over.
Yep.
The other important thing I forgot to mention with the other ones is to try and get rid of the residual water in the filter. So at some point you need to push a syringe full of air through the filter, either by hand with a caulking gun, or just lift this out of the water and then just push it through and get rid of the water in there. Then the idea being is in fact, the way that you do it, what we want to do is get this preservative onto the filter. And so what we're doing is the technique is to take that little cap off, put it on there, unscrew it from there, pop it on there, and then you've got your syringe and your filter, and then push the preservative onto the filter, and there'll be a bubble in there, and that's okay. So don't try and get rid of the bubble. The main thing that happens with that bubble is that everyone squirts it out and you lose the preservative. So put it in, push the bubble on. I won't do it this, otherwise it's going to be all wet. But yeah, and then that is how the sample stays.
Yep.
And so you ship that.
That's your complete sample ready to go off to the lab.
Yeah, yep. And so that's water sampling. So there's a few considerations we can talk about after for that. So switching across to rollers, I guess the same thing applies. This is a roller kit. We've just got everything in there. Again, first things first, take out your set of gloves. And the next part of this process is, so we ship the preservative in a syringe. We used to ship it just in this little holding tube, but once they get into airplanes, they leak. And so that wasn't going to be any good. So we've got, in these, they seem to be much more secure. And then basically what you're doing is putting that preservative into this tube ready to take your samples. So no need to demonstrate that. But then the next part is we've got these little rollers in the autoclave bags. They're sterile, ready to go, with a little black hole at the end. You'll see these in Bunnings. So these are something nice and simple. They're microfiber rollers, so they pick up surfaces really nicely. This is a little sampling handle, and what you're doing is popping that on there and putting it on. And now, if you're sampling like a shallow water where you can't filter water, there's a lot of groups that look at puddles, for instance, then you can use that straight away and roll the bits on there. I guess the point when you're doing that is try not to get this too wet, because when it's super wet, it's just going to dilute the preservative. But when you're out, the whole purpose of this was when you're out in somewhere that it's dry, and you want to do woody logs or bushes and stuff, then we also get you some clean, sterile water here. And the idea is just to get it a little bit wet. And you don't have to soak it, just make it wet, because it just seems to pick up stuff a little bit better when it's like that. And then you're going to roll it across the surface. Aim for a square metre, for instance, or what you did was very small, so yeah. I'm not sure if we got any data out of that.
Probably not.
I don't think we didn't have any fails, so we did. So yeah, there was something out of that short roll. But when you're rolling, there's not much point in rolling the same space again and again, because basically you've taken the surface signal off that. So roll somewhere different and just keep going and time yourself for a minute, or aim for a square metre or something like that. And then that's your sample. So then what you're doing is, this is full of preservative. Then you're just popping it into the tube. Try to get this technique going where you can take off a lid with one hand. You're popping it in there, and then you just have to, because it's a bit tricky to take it off, you put the lid over the edge, and then you're pulling it out like that.
You've done that once or twice before.
I've had some practice.
Yeah.
And so that's your sample. And so you don't have to, you can shake it if you like, it doesn't really matter, but what ultimately happens is the preservative works its way up the roller and preserves it. Might not get to the top, but that doesn't matter, because we're preserving the bit that we can get to. And then that's good for us, so you'll be able to seal it up. I think we give you some paraffin to seal the edge, but largely that preservative is soaked into the roller and we don't have to do too much more there. The other one was air. Well, I can do.
Yeah, I think air is pretty straightforward, and I have got a question on soil sampling.
Sure.
So let's talk about
So soil sampling, same thing again, just a kit. And I guess this, you've got a tube and you've got a little scoop. And I guess the way that we've tried to design this is, it's good if we also give you a big Ziploc bag. So I'm not sure what the volume is, but what you can do with soil is, this is your little scoop. You're pulling it out and take little scoops, put it into a Ziploc bag. You can do from a larger area. and then just shake your bag, and then take that, scoop, and then you're popping it in there, straight in, and closing the lid.
Yep.
So that'll fit in there. And I guess we don't want too much because we actually do the extraction in this tube. So we've got a little line in there that sort of don't go above that line. And this is true for scats as well. So if we get, these will be the same kits for scats. We don't need all of the scat. Prefer we didn't because it becomes gross. But that's enough for us to take. Now, unfortunately, preservatives, we haven't come up with a good preservative that properly stops everything. Scats aren't too bad when they get dried out. We've had lots of scats that have been out in the environment, just sitting in the sun, drying out. And they've actually worked fine. So I guess scats, if you can dry them out, then you can pop them into a paper bag or a Ziploc bag or something. I guess a Ziploc bag actually keeps the moisture in, so that'll keep the bacteria animated. So I guess paper bags, or at least dry, is better for those guys. But soil is better frozen, and so if you can freeze it and keep it frozen, and as long as it's not going through too many freeze-thaw cycles, because that tends to animate bacteria, then that can work well.
So from the field straight into the freezer, and then as quickly as possible. Or
in an esky with a bunch of ice blocks for the day, and then into the freezer.
Yeah. And then
you get back
as quickly as possible to the lab.
Yeah.
Yep. So it doesn't matter if it's in your home freezer or your work freezer, and then it's going to thaw between that time.
As long as it's not a full thaw, yeah. I mean, we get a lot of coral samples that are frozen off site with biofouling, and the outside thaws a little bit, and that's okay. But there's usually stuff on the inside that's still nice and well preserved.
Okay.
Because ultimately, if it thaws, bacteria will eat it and it'll go away.
So another question we've got here, Shane, is about bleach and decontamination of equipment between sites.
Oh,
yeah.
10% is, and it's 10% active bleach, and there's a calculation we need to do for that. We use the little bleach tablets from Coles. I haven't seen them in Woolworths, but they're little White King tablets, and we use 2 of those per litre. And so that active bleach is above 10%. And so, I mean, more, 20% is still okay. I guess 10% for 15 minutes, I think, is the, just my memory here, I might have to follow up and confirm on that one. And that will properly bleach, kill everything. 20% is okay too. But just both of those, you just need to make sure you rinse that bleach off, because you obviously don't want it on your equipment when you then take the next sample.
Yeah. Okay. And then the next question is about minimum soil sample mass, which you did touch on.
Actually, I've just had a thought on that.
Yep.
If I can just follow up to that one. When out in the deep sea and on lakes and things, decontaminating things can be really tricky with bleach, and it's particularly out in Pilbara in many cases. Bleach is, you can't just pour it on the ground, on the tenement rules, etc. So dilution is not a taboo topic. So if you need to clean something, rinsing it underwater, there is a technique, I think it's for 3 minutes or something. I don't know if you remember that one. Again, I'll have to follow up, but if you rinse things thoroughly, then in your new site water, then you've replaced everything that you've brought to that site, and you've diluted all of those signals down to very low, and then you take a new sample with the new equipment. It's a compromise, but this is what has to happen out in the field.
Okay. All right, so this question of minimum soil samples, you touched on it for your kits, but to this question here, typically...
Yeah, only using small. Yeah, I mean, this is true for all samples. So you start with as much as you can, you're homogenizing it, you're subsampling it, you're extracting it, you're subsampling it, everything is slowly getting smaller and smaller. So what our suggestion is to take, I mean, we don't necessarily have an amount that goes into the Ziploc bag, but if you take your little spoon, or you get a square meter, let's say, and you take lots of samples, or it could be bigger, it doesn't matter, put it into a Ziploc bag, and then homogenize that, and then you take that little spoon, that's your subsample of that set. And this is what we mean about trying to keep your methods as consistent as possible. If you do it once, one area, and you need it to be comparable, we'll do it in the next area, and you need it to be comparable. But that concept of using less and less and less, it happens regardless of what you do in the sampling side. So yeah, it's good to try and capture a good area to start with.
I do notice too that on your red line that you have here, it says 25 mil, which, I mean, roughly, depending on the density of the soil sample, is going to be about 20-25 grams, isn't it?
Yeah.
No more than that.
Yeah, and I mean, that's more of a practical concept as to what we can accept. So if you fill that up halfway, then we can put all our beads that we bead bash the sample, knocks everything apart, and then that homogenises that, and then we're subsampling from that to do the extraction.
Excellent. Question here about your preferred biodiversity index, Shane.
So we haven't got a specific biodiversity index. We're representatives of Woodlab over here. They have a taxonomic independent classification index ticking, which we can apply across that data. Angus's work is working towards a similar concept, but based on reference data specific to Western Australia, and that's called aquatic, which is more or less the same thing. And yeah, so those are those indexes that we can work with you on. But that's part of the research, and Marina's work is looking towards generating a terrestrial index. I know there's a lot of macroinvertebrate scoring systems, but that's not something we work with.
Okay. We've got any more questions there? Can we get an indication of testing kits and their broad? Yep.
eDNAfrontiers.com.au. Everything's on our website there. There's a little tab that talks to sampling. There's lots of information there. Otherwise, you're welcome to make contact, and we'll get all that information to you.
Very good. All right. No more questions in the column there, Thomas. We're all done. No last minute. Here we go.
Water sampling coupled with water that has high sediments. Yeah. Rangelands red. That's special. It does tend to really clog things up pretty well. So we do a lot of, as an example, I guess, we do a lot of Pilbara olive testing in rock pools up in the Pilbara. And that water occasionally is super red, and those filters come back as red as they get and chock-a-block. So yeah, you're not going to get a litre out of those samples. We try and use different size filters to make sure we've got something that works, but typically we don't go above 5 microns. 1.2s generally get enough of the sample to be representative from that area. But you're never going to get rid of that sediment. It's allowed, shall I say. If you take your litre, if you've got a nice clean water container, you dip that in the water and let it sit for, however long that is sensible, 5-10 minutes or something, and then take your sample from there, that's okay. Not all of that sediment's going to fall out of the water. It's going to stay there a lot. But the bigger bits will. Anything that's got some gravity to it is going to fall out, and that just helps clean it up a little bit.
Yeah, it gives you an opportunity to get a little bit more volume through the syringe.
The filter will block up eventually anyway, and there's no way around it. And I guess you don't want to push it too hard, because you do want to try and get rid of that water at the end and get the preservative onto it. So go as far as you can, but as soon as it starts blocking, then just stop.
Yeah. Excellent. Well, I think we are just about finished up for the morning. I'd like to thank all of the participants. I'd especially like to thank Marina and yourself, Shane, for your presentations. Just to remind people that we will be putting a recorded version of today's presentation up on the RegenWA website, so go searching for that. And also, there is more information on the RegenWA website regarding this broader project, including a video that was made post the workshop on the 5th of May. It's about a 12 or 13-minute video, which is a very good overview. Shows Marina and Shane and also Angus, Laurie, their presentations there, but it gives a broader background to the overall project. And keep following us, because this project's got at least another year and a half, nearly 2 years to run to its conclusion. So we are moving to the implementation phase now that we've done the testing and identified some ecologically endangered / threatened species on the properties that we tested. So there'll be some on-ground works happening later this year, and then we'll be going back to retest and see what impact we can have in a positive way.
See what's changed.
Yeah, exactly. See what's changed over time. So thank you once again, everybody. I hope this has been an informative webinar and use of your time this morning, and very much appreciate your attendance. And have a great day.
Cool. Thanks, everybody. Thanks for
you.
joining.